Welcome everyone, it is Mr. Olson. Today I'm going to be walking you through the bacteria lab that we ran today. So today, realistically what happened is we plated a bunch of different bacterias and we're also plated antibiotics on top of those bacterias to see what antibiotics work best with which type of bacteria. So, first thing I gave all the students today was a Petri dish. Now this Petri dish, so let me get this fire out the way. This Petri dish is quadranted off, labeled 1, 2, 3, 4, and also has a layer of nutrient agar labeled in the bottom. Now, nutrient agar, realistically what it is, it's a Thanksgiving dinner for all of the bacteria. So it gives them enough food to actually be able to grow. Now, this will also be going into an incubator, so the incubator puts it at a prime temperature, allowing for bacteria to grow at an optimal rate. So, today, we're going to be working with a couple different things. We're going to be working with, one first and foremost, a Bunsen burner. We're going to be working with an inoculating loop. We're going to be working with something called a slant slide. And then we're going to be working with antibiotic discs, which I'm going to show you guys in a second. So first thing, we have an inoculating loop, which is just a big wire with a little circle in the front. Okay, I hope I can get that focused and it looks okay. So what I'm going to first do is I'm going to grab this fire and I am going to heat up this inoculating loop. Now the goal of this, guys, is to get this to turn bright red and just kind of keep going back and forth until the whole thing gets bright red at some point. What is happening here is that this is actually disinfecting or sanitizing our inoculating loop. We want to make sure that the bacteria that we plate is the bacteria we want there. We don't want any cross-contamination. And this is kind of our way of doing this, or is our way of doing it. So, heat it up, get glowing red. Glows pretty red, pretty good there. A little bit farther down the base. Bam. Now, I'm going to take my inoculating loop. I'm going to take my Petri dish. I'm also going to take this slant slide that also has agar in it. It has bacteria on the surface. I'm going to use my right hand. I'm going to uncap the slant slide. I'm going to use my inoculating loop, and I'm going to go inside. I'm going to gently scrape off the top layer of this agar. Trying to collect as much bacteria as I possibly can. Rotate it. Do it again. Do it again. I'm going to cover this back up. The bacteria I'm actually plating today, guys, is E. coli. Okay? I'm then going to go to my Petri dish. On my Petri dish, I'm not going to actually open it all the way. I'm going to try to keep all the contaminants out. I'm going to make big S marks all the way across. Now I'm going to rotate it. So hopefully this means I'll get every surface on here to have optimal growth. Do it again. Just for good measures, guys, I'm going to rotate it one more time to make sure this plate actually turns out really good. Bam. Close it back up, flip it over. Now this inoculating loop has bacteria on it, so I have to make sure it gets clean again, so let's just reheat it. So we're just going to put it back under the fire. We're going to reheat it up. Get it back to glowing red. Perfect. It's done. Put it down. I'm going to turn off the fire. Beautiful. Once the fire's out, I'm going to take my goggles off. So now I'm safe. So, the big thing with this lab, guys, is that we're going to be testing different antibiotics. Now, antibiotics, as we know, they kill bacteria. But why do we take different types? Why do we have different antibiotics for certain bacterial infections? Or why do we have to keep making new antibiotics? That's kind of what we're going to get to here. So, there's bacteria on this plate. Now, this is E. coli, so we're going to see what type of antibiotics actually kill it the best. So, we're actually going to look at 4 different types. We're going to look at streptomycin, erythromycin, tetracycline, and neomycin. The first I'm going to plate is going to be streptomycin. Now, our antibiotics come on little like white pieces of paper that looks something like this. Okay, and it's even labeled with an E. So what I'm going to do is I'm going to take this Petri dish, I'm going to place this in quadrant 1, gently push down on it. I'm going to shut it back up. And now erythromycin, or streptocycline is on there, my bad. Streptocycline is on there. Now, number 2 is erythromycin. We'll grab that antibiotic, just right here. Grab this antibiotic. Do the same process. Grab the disc, go to quadrant 2, place it on there, gently press down, bam, that's done. Now the third one is tetracycline, which I'm gonna grab again. Grab this over here, grab my antibiotic, put that in quadrant 3, press down. Goes there, goes there. My fourth and final one, that's an antibiotic, is my neomycin. Push that in there. Beautiful. So I should have got something that looks kind of like this. Okay. I have my 4 antibiotics with my bacteria. Now, I'm going to plate one more thing. I'm going to plate something in the middle. It's going to be called our control. Now, these pieces of paper are just filter paper. There's no antibiotics on it. So it should prove that the antibiotics are what killing the bacteria, not just the presence of paper. So what should happen is that the bacteria should actually grow right up until the control and just grow right around it, showing that it doesn't kill anything. Is that going to work? I don't know. We'll have to wait to see till next week. But it's kind of the idea behind using this control or using this piece of paper as a control. So I'm going to put this piece, put it right in the middle. Do my best to get it down there flat. It's on there pretty good. So now I have those 5 pieces. So just a recap of what we did. We plated bacteria, we plated E. coli from our slime slide using our inoculating loop. Now we did everything sanitary by using fire to burn it, so using our Bunsen burners, which was important, so we don't want any cross-contamination. I then plated my 4 types of antibiotics. Plated streptomycin, erythromycin, neomycin, and tetracycline. Then I also plated a control disc just to prove that, to make sure that those antibiotics are actually doing their job and killing the bacteria. And we should get something that looks like this. Now, in our experiment, guys, E. coli actually comes out to be B for our experiments. So I'm gonna make sure I label this with a B before I put it in the incubator, which the incubator will be at 98.6 degrees, giving it optimal temperature for optimal growth. Other than that, guys, that's what the lab consisted of today. We'll see what the results look like last week. I'll talk to you guys later. Bye.